interest slide oh, recording. Thank you so much, whoever, Jasmine. So Anabel is a computational engineer whose interests lie in the intersection of applied mathematics and software engineering. She received her doctorate in chemical engineering at Oklahoma State University, where she conducted research in the field of computational biosystems. Her current research work is in NNDescent.cpp, a cache efficient and distributable reimplementation of PyNNDescent. And recently, in December of last year, she joined Stellar Science as a computational engineer. So we would like to extend a warm welcome to Anabel for giving a talk with us today. Feel free to take it away.
Thank you. But yeah, I'm going to be going over some dimensionality reduction methods. Primarily going to focus on PCA and UMAP as two relevant entry algorithms into this topic. And before I go too far into this, there is a couple of caveats to this. First, my background is in engineering, not software engineering, like chemical engineering, as you mentioned, not mathematics. Some of the formalisms of this topic do escape me. I mainly just know PDEs and how to solve them. Okay, not the only thing, but that's what was taught to me. And I'm not an R expert. Most of my experience has been in other programming languages. And so there's going to be plenty of you in the audience who know more about R than me. I actually earlier today made a joke about, oh yeah, programming is just a constant state of looking at your code written 6 months ago and cringing. I looked at this code that I had written for this about like 6 months ago, and I'm like, oh God, some of the names in here are terrible. But my R, good God. Couple more caveats. My focus is to present a mental model for what these algorithms actually do. These are conceptually implemented as functions. There are different parameters that you can put into them, different options. The built-in help does help and describe a lot more about the different options you can do with them. My perspective is that is much easier to use, like the help is much easier to use, and using the algorithms are much easier when you have a good mental model for what's going on. And I'm re-giving this presentation. I have a lot more time this round. Please stop me, ask questions. I am abstracting over a lot, and maybe the abstraction I'm doing is wrong for you. That's fine. It could also be I'm just not explaining something that I should have. And please, if I'm doing that, yell at me. But why do we care about dimensionality reduction? There's first visualization. We've got this data that only makes sense to think of it as, I don't know, 600 dimensions. And we want to actually be able to see it, because us humans don't really know how to think about 3. And so these are ways to actually meaningfully take high-dimensional data and represent it in some way that makes sense, in a way that we can see and visualize. Another aspect is that it can be a part of a chain of models where a lot of the modeling and computations might be computationally expensive and directly depend on the size of the input coming into them. This was from like a really toy side project where I was doing stuff with neural networks and convolution filters. And that's the type of thing where with the convolution process, you do want to actually reduce the size of the image as you can evolve into more and more features. And that because keeping the dimensions down helps to not burn out your CPU trying to just run something and end up taking 5 hours to do it. And the example I have going through that I have associated with this is actually something I used as a real example for me. And so I mentioned that I'm working on this algorithm, and part of it is that at the time I was using this data set MNIST fashion, which is kind of it's a good data set to test like some prototypes against. It's actually a spinoff of the MNIST digits database. But MNIST fashion is a data set that has 60,000 images in the training set, 10,000 images in the test set. There's 10 different types of clothing, so 10 different classifications, mirrors the MNIST digits 0 through 9. And it is 28 grayscale images, or 784 dimensional vectors. And this is what it looks like when you apply UMAP to it. I actually don't have the clothing classification on here. And part of it is
Could you
because
share your screen with us?
I am a complete idiot.
I
Oh my God.
No, it's okay. I didn't realize until you were, maybe we back up a bit. Yeah.
Okay, so
Thank you so much. Sorry.
okay, so
I
this
catch it.
is this was what I was talking about, the modeling. And this is the data set, and I am so sorry about that. And so this is what it looks like when you apply UMAP to it in this fashion. And part of what I wanted to see out of this was nearest neighbor descent, the NNDescent, where you get NNDescent in both what I named the work name for my project and PyNNDescent. What PyNNDescent is spatially partition all the data points into distinct sections of the space. And okay, now it's not showing people's cameras. And then it just does that, forms graphs in each of the partitions, and then does an initial random initialization over that. What I wanted to do was say, hey, I could think of these as separate blocks, figure out like a centroid for them. And instead of just doing a random initialization to figure out what other points in what other sections are neighbors with each other, I can first try and compare different areas of the space that might be closer to each other first. And so that's what I ended up doing here, where I projected UMAP in two-dimensional space, but then I also projected the centroids down and try to see where connecting the centroids. In this case, I'm connecting the nearest 3 centroids to each other. And there's a bunch of lines going across clusters. That's expected, that's normal. But there's also a large number that are within clusters. But a quick diversion, I'm going to start writing some code. At least on my computer, running PCA takes a bit of time. So I'm just going to start this now so I can come back to it a little bit later. A little bit later in the presentation. And to first, I'd like to start by talking about very top level, 2 different types of techniques. There's linear dimensionality reduction techniques and nonlinear. And with linear, well, easy. Okay, easier. It's not really that easy, but relatively speaking. Okay, faster. Same thing. It's not necessarily a super fast thing, but relatively speaking, it is faster than nonlinear. And it can become lossy. You can lose some of the structure in your data that you actually care about. And under hood, it's a matrix problem, typically some sort of matrix factorization problem. As for nonlinear techniques, they're harder. Both the theory and computation is much more complex, takes more time, but they typically aim to preserve more structure. It depends on what algorithm you're actually using, what aspects of that structure they preserve best. But under hood, they're mainly some sort of optimization problem. How do we take this lower dimensional layout and more closely mimic that bit we want to preserve to what's present in the higher dimensional data? And so bird's eye view of linear dimensionality reduction. I mentioned matrices. Well, what we can do is take a data set. This could be experimental data where we have different variables that we're measuring, or it could be like with in this fashion, images where the columns are individual grayscale pixels and the rows are individual images. And so we've got this matrix of experiments by variables. And we want to take this and preserve the rows, but reduce the number of columns we're working with. Take our individual entries and represent them with something with a much smaller amount of data. I do want to note, calling them real numbers when we're dealing with computers, a bit of a lie. If you want me to expand more on that, I can. Just something to keep in mind. But principal component analysis, PCA is all about describing your data in terms of the variance within that data. And with this two-dimensional cluster, the first line, it goes from bottom left to top right. And then from there, we figure out, okay, orthogonal to this line, what is the next line that we can draw that best describes the variance in the data? In this case, we're constrained because 2 dimensions. But if it was in 3 dimensions, we'd have an entire plane to work with. In well above 3 dimensions, in n dimensions, we draw our first line. After that, we have n-1 dimensions to work with. And so once we've drawn those lines, we can then reorient our data, remove the original coordinate system, and replace it with our lines of variance, and then take the axes of variance that are the least significant and project everything else down onto the more significant ones. In this two-dimensional case, it'd be conceptually equivalent to just collapsing that second dimension and projecting everything straight down onto the line. So DSVD, because this is very briefly the canonical form, where X is some square matrix. U and V are orthonormal projections. Orthonormal projections are rotations, mirroring, kind of like going from here to here. That part, that is an orthonormal projection. All the points maintain their distance relative to each other. And here's why I completely switched over to talking about the SVD. The SVD, that sigma matrix, that's a scaling matrix or diagonal entries, where that's the eigenvalues of XCX. XCX, that's the covariance of our data. PCA cares about finding the eigenvalues of the covariance matrix. In other words, this is a closed form solution of PCA. Not only is a closed form solution of PCA, the SVD is basically an emergent algorithm for solving this, basically what amounts to the same problem in a wide variety of disciplines. So this is the grandfather of all dimensionality reduction and data compression. As for the thin SVD, well, that's a bit more relevant here because we don't need to care about having a square matrix. We can have different number of columns and rows. And we can think of V as rotating the axes from the origin to the lines of variance in the data. And then just get rid of the X and V. This is what we have, where U, this is going to be the same size as our data matrix, and each column has magnitude of 1. Kind of like creating a new basis for our system instead of having a vector that is 1, 0, 0, 0, 1, 0, 0, 0, 1, for normal Euclidean, a normal coordinate system. These serve as the new bases for our data. And this matrix, well, this represents how significant each of our lines of variance are. And some of these entries may be 0. In other words, free dimensionality reduction. Okay, that pesky little thing about not really working in floats comes into play here. you're going to get error, and you're going to get values that might be really close to 0 that you can just say might as well be 0. But how can this be used for dimensionality reduction? Because now we have more data. We have a matrix that's the same size as what we started with, and now this diagonal matrix. Well, one of the properties about the SVD is that the diagonal matrix, its elements are in by magnitude strictly descending order. The sign can change, but the absolute value of the 0th element bigger than the first, bigger than the second, bigger than the third, so on and so forth. So what we can kind of conceptually do is take the least significant elements and just lock them off, just discard them, cut them off. And then we end up with U. We've got same number of rows, 2 or 3 columns if we're trying to visualize stuff. Maybe we're going not from 784 to 2 or 3, but from 784 to 50, just to make the data more workable with for some other computation. And let's explore what our code gave us. So this is what the data set looks like using the block numbers from my algorithm. Essentially, things that are in the same block are in the same region of space. And we can see we do get this kind of nice gradient out of it. But we also had 10 different types of clothing. And there is some segregation of what's going on in there. But then the rest is just smeared out across the image. And this is...
Hey, Anabel, can I ask
sorry,
a
yeah?
question?
Yeah.
So if I wanted to follow what you're doing here, which file can I refer to in your GitHub?
Like I mentioned, some of the naming is this is bad. This is PCA.R. You do need to source it because it does pull in, it does call this other file that I have that is just reading in from the binary data. Okay.
And the other request is, can you make your font a little bigger? Because from my screen, I have a hard time seeing what the code is. Yeah, that's better for me. Can everyone else see it?
That's much better from my standpoint too.
Okay.
I do have a few other images to show. Oh God, that looks terrible now. I do have a few other images to show. I can readjust as I go.
Thank you.
So built into R, there's 2 functions that will do PCA for you. There's PRComp and PrinComp. That's the names that was decided for them. PRComp does SVD. PrinComp does explicit formation of ATA, and then does eigenvalue decomposition on it. So I expected the SVD to be faster than explicit eigendecomp. It turns out only a few seconds faster in here, but it is definitely less error. So I do strongly recommend just using PRComp, just using PRComp and not PrinComp. And I even think if I remember correctly, going into some of the help documentation does even recommend use this, avoid using this. But here, I have my data set represented as a matrix. I believe you can use a data frame. Once again, this is one of those things where I'm not very familiar with R's data frames. I'm much more familiar with matrices. But this PCA, this is where I'm getting the results out of PCA. And so some of the things it will give us is, so this gives us some standard deviations of our data. And it's explicitly this X value that I'm printing out into this plot here. This is the data with the dimensions reduced. As for the rest of this stuff, well, part of the reason why I chose the example I showed is not only visualizing this, I'm also taking data that's outside of the set, derived from it really, and using the same transformation to project it. That's this next set of lines here. It's a bit noisy, but essentially what I've done is take the rotation matrix and multiplied my centroids by it. Multiplying the centroids on the right by that rotation matrix will produce a reduced version of my centroids. And let's see if this is going to cooperate now. It wasn't earlier. It was taking its sweet time to print stuff. Oh no, I look like a complete fool. What went wrong with my code? Yeah, okay, I'm making a point here. So one of the things that I kind of showed before with transforming the coordinate system, when I removed the origin of the new coordinate system didn't line up with the origin of the original data. And one of the things that PCA does is first centers the data on the average and then does the rotations on it and rotations and scalings. And so what you have to, that's what this center is for. So what you first got to do is actually take your data, subtract the centers from it to center it in on the new coordinate system, and then do the multiplication to rotate it, or well, in this case, project it. And so so now that I've taken account that translation, I've now successfully projected the centroids onto the same coordinate system, and can kind of see here that I had looked at the direct numbers. Okay, how many neighbors in this block line up with our neighbors with points in this block? But like seeing this kind of gave me more of a glimmer of hope in like, okay, this is something to actually explore. But what about UMAP? Well, I'm going to go over to this other file, start run. Oh, I'm going to first clear out my work. Before I actually do, I'm usually clear out my work environment. I like always running my stuff with a very fresh, clean environment. Before I go any further, does anyone have any questions about this?
I just have one question. So do we need to actually standardize the data first or just subtract the mean that it will be enough?
So PR comp and print comp already take care of averaging out, like centering your data on the average. That's taken care of for you. As for normalizing it, it does not normalize it. The SVD doesn't care if your data is normalized or not. You can if you want to. I don't know about, I'm not sure about like the explicit eigendecomp. I wouldn't think it would care if it's normalized or not first. And so if you want your data to be normalized first, you would want to normalize it first yourself.
Okay. And another question I had was, so you mentioned that the processing time for PR comp and print comp was different, but will both of them give the same result?
So
I mean, like if
so the time isn't exactly different. I'd expected the SVD to be faster. It might have been an implementation detail, but they are the same closed form solution, but how they get there is different. And how the SVD gets there by gets there while accumulating less computational error. I can explain a bit more about that. That's kind of the very glossed over warning about not working with real numbers that I mentioned. I can expand upon that a little bit further, but essentially every operation has some sort of cost at a, if you're working with double precision number, 64-bit numbers, 1 to 10 to the minus 15th relative to the actual magnitude, you accumulate some sort of error every time you do an operation. And when you chain operations, that compounds and builds.
Okay. Okay. Thank you.
Mm-hmm. And SVD just compounds less error over time.
Right.
All right. So I'm going to clear this and start writing the stuff related to UMAP and go back to the presentation and wrap up some of the PCA stuff. And source. So I talked about PCA in terms of matrices and rotations and whatnot, partially because that's how I knew the SVD before even thinking about it in terms of the PCA. That was something I was a bit more familiar with, but you can take the same concept of basically matrix factorization and think of it a little bit more abstractly, a little bit closer to the data actually. And think of it as trying to find a set of reduced columns, which are amounts, and column 1 represents some sort of feature, like magnitude of a feature, and column 2 represents a magnitude of a second feature. In the case of SVD, these features you get are, in this case, these would be linear combinations of 3-dimensional space. Feature 1, 5 steps along X, 4 steps along Y, 6 steps along Z. And you can define from these 2 features a 2-dimensional system. But we don't even need to work with real numbers. We could have some sort of ordinal thing, like we have responses that are none, a little, some, a lot, all the time. And it wouldn't necessarily make sense to apply PCA to that. PCA is reducing the residual squared. We might want to apply some sort of different error function to it. And going down that route, there's generalized low rank models by Udell. There's also a presentation that I have linked later on the slides, but her work goes into that concept. But even then, the first step of those algorithms are just SVD it, because we get pretty close with that as a starting point. And so to recap, PCA it's fast. Using big O notation, it's m times n squared, where n is dimensionality of the data, and m is the number of data points in our data set. In other words, adding more points to our data set yields a linear increase in computational time. And that's not bad, that's pretty good. And if you're lucky, it can just reveal inherent lower dimensional structure in your data just for free. You just get it. But beyond that, you lose information. You lose the representation of your original matrix. And with that, I'm
quick
about
question
good time for
regarding that the 3 points that you just made, if you could go back. So does this mean that the more data points you have as input for your PCA, the better chances you'll kind of detect any like lower signals or lower I guess factors that don't have such a big impact on your data set?
Not necessarily, because that's going to be a property of what's actually producing the data. If you're thinking getting that from the first point, what that has to do with is if you have 60,000 data points and it takes you 2 minutes, you can expect 120,000 to take 4 minutes. Really, this is asymptotic complexity, which means, okay, as you go into infinity, how does it grow? But that second and third point, those are properties of the original source of the data, essentially, not how much of it you have. Granted, if you have more of it, you might have noise that becomes less significant because you can you have more points to actually get what the actual underlying structure is. So there is something to say about like the errors of your measurement, the errors of the data you have itself, not computational error, but like experimental error. It's been a while since I've walked through the like specific math on that. I'm a little hesitant to say too much. Generally, I'd assume if you have noisy data, you would want to have more, but there are cases where that doesn't help and you just need to reduce the error to first place.
Right. So I guess like I'm coming from, I'm coming from a geneticist point of view. So my primary role is in cancer genetics. So when I use PCA or think about PCA, I kind of think about I have all these genetic data points and what are the biggest factors that are influencing different groupings of my data. And usually sex is one of the biggest drivers of genetic differences. So something like sex is something I can see immediately in my principal component 1 and 2. But I was wondering, like, so your second point is saying that if you're lucky, it reveals inherent lower dimensional structures. So like that would be other drivers that don't have such a big impact like sex. So for example, if it were specific genetic mutation that was driving differences in my phenotype data, for example. So if that particular driver doesn't have such a big effect, does this mean that if I collected enough data and this driver, whatever it is, the effect is a linear effect, then having more data points would help detect that difference?
Yeah, I guess it could help detect it if the data is noisy. I also, part of it is that you're gonna want more rows and columns or more experiments than you have of variables. And yeah, but essentially it can pick that up. Yeah, this gene is not really a thing that matters, but it won't explicitly say, oh, this gene is a thing that doesn't matter. What it will do is say, oh yeah, this diagonal matrix, you are expecting, let's say 50 elements, but only 45 are non-zero, meaning the actual data, was essentially produced from 45 genes if we ignored like nonlinear effects, which I am ignoring nonlinear effects right now. And so it won't tell you what 45 genes matter. Just you could, in theory, trudge through all the numbers and figure that out. But what you will explicitly get looking at the diagonal matrix is, oh, only 45 things matter here.
Okay, I see. And when you say it will destroy information that can be teased out through nonlinear projection, so that means that PCA is really suited for linear effects.
Yeah,
so that doesn't fit that particular model. Isn't detectable using PCA?
So to a degree, yes. So part of it is that the SVD is a very, very good at figuring out how things linearly relate to each other. In fact, I kind of glossed over it, but it's a closed form solution to the linear least squares problem. Essentially, what are the difference of the residuals or the residuals squared between like, kind of like my model and what the data is. Because like I'm trying to draw a line through it, I'm trying to fit some model to it. It solves that for matrices essentially. And so when you have, in the case where you have the full SVD, where you're not removing dimensions, this right here, this is in math land, this is strictly true. These factorizations is an exact representation of the X matrix. And part of that is figuring out, okay, what's the two rotations and this scaling that I need to figure out to represent this matrix. And as for the losing information part, as you start removing non-zero elements of the diagonal matrix, this on the right-hand side becomes a progressively worse and worse representation of X. In other words, once you remove the first non-zero element, this equality does not hold true. How much it holds true depends on the relative magnitudes of the sigma matrix and the relative magnitudes and how much and how big the elements are, how many elements you cut off and how big those elements are. And essentially what you end up happening is if you multiply the reduced version of X, or multiply a vector by the reduced version of X, you can actually measure, okay, what's like the residuals, what's the difference between the residuals squared? And in the case of square matrices, you can get a solution that has no difference. Does that kind of explain, or did I completely miss the question?
Oh, that's good. Thank you.
All right.
Can I ask a question before you get going again?
Mm-hmm. Sure.
So one of the, I approach this much more, I'm very familiar with the mathematical side of the of dimensionality reduction, but another way that we tend to approach this is as a way to make data more interpretable. And that's largely, especially in the biology space and things like that, you're looking for separations of clusters and things like that. I don't know if you're going to get into this when you talk about UMAP versus PCA, but I was noticing that your PCA just really did not show any sort of additional dimensionality features after you had run it. Your data was very centralized. And so I wasn't sure if you were going to speak to the difference between PCA and UMAP and tSNE or things like that with regard to the interpretability of the outcome versus just the mathematics.
So as for I do touch on that a bit later. I do touch on it more briefly than may have been ideal. But in this case with the PCA, or with PCA, the actual dimensions that you're projecting against or down to the visualization, the axes do have like actual physical significance. They're essentially linear combinations of the higher dimensional data space. It might be like, oh, this one axis is the kind of effects of these genes to this effect, and another axis might be this other potentially overlapping set of genes to this effect. And so with PCA, you get stuff, like one of the good things about it is that you do lose representation of your data, but the actual coordinates you're projecting them onto have some sort of real physical meaning. With tSNE and UMAP, you don't get that. It does show clustering. It does show like things that might be more related to each other. And in the case of tSNE and UMAP, they both try to preserve points that are close to each other in the higher dimensional data set, but a lower dimensional space, no real physical significance.
Right, right, but it's more interpretable. Okay, yeah, excellent. I was just
Yeah.
wondering if you were going to touch on that. Thanks.
Yeah, so I do touch on that briefly, not a whole lot, and it's good that you asked because there is a bit more nuance to talk about there. I expanded upon it briefly, but yeah, that's PCA. While I did mention like it loses more information, the coordinate system, the axes makes sense. UMAP, tSNE, you lose the meaning of the coordinate system itself. Really brief recap of that.
Absolutely.
Just summarizing for not so yeah, but yeah, no problems asking questions. You know, especially right now because I'm about to transition to another topic, and this is like great to ask me to go back now before I leave this topic behind, this portion of the topic behind. Anything else? All right. Okay, so bridging into nonlinear dimensionality reduction. I mentioned it was an optimization problem, and in the case of tSNE and UMAP, the optimization problem that we're doing is optimizing probability distributions. And so, really brief bird's eye view. In our higher dimensional data set, we have points. We can calculate distance between them, and we can map each of those distances to a probability distribution. Similarly, we can take our lower dimensional layout and do the same thing. Take all our points and map their distances to a probability distribution. The goal? Reduce the changes, reduce the difference in our probability distributions, get them to line up by tweaking the positions of all of our data points in the lower dimensional space. Once we get the probability distributions to line up as well as we can, we have something that should be a good representation of our data. And so what tSNE does is says, let's fit this stuff to a Gaussian distribution. And so for each of our distances, this is the computation it does. Fits it to a Gaussian distribution. And I mentioned that PCA itself was linear with respect to size of the data. This denominator, the best we can do for this is approximate it in order n log n time. So this is already super linear, and there's other complexities going on here as well. Including this is just one point. We need to do the entire data set. And well, complete tangent, not related at all. Okay, this is related. K-nearest neighbor graphs. K-nearest neighbor graphs, for different reasons, different ways, do relate to nonlinear dimensionality reduction. In the case of tSNE, this is just an optimization. Say we only care about calculating the probabilities for a point to its k nearest points. And this is conceptually equivalent to taking our Gaussian distribution and just cutting off the tails. Focus towards the center of the bell curve and where most of the area is. Well, what is UMAP then? At a high level, and glossing over a lot here, it looks a lot like tSNE. First step, form a k-nearest neighbors graph. Next step, compute dataset probability distribution. Third step, optimize probability distribution of the lower dimensional layout. However, the mathematical theory behind it is a lot more complex. tSNE does quite a bit of hand waving. UMAP does a lot less. And part of that is that it gets out a probability distribution that A, makes sense, and B, much faster to compute, much faster to optimize in the lower dimensional layout. And so we can kind of think of UMAP as trying to do surface approximation with our data. Take a three-dimensional graphics model, and typically they don't necessarily need to be only just triangles, but classically the example is representing a three-dimensional object, but as a two-dimensional series of triangles in this three-dimensional space. And that's kind of what UMAP is doing, kind of like conceptually forming this triangular mesh, except instead of a three-dimensional space, it's like, 784. And the actual name of it is Uniform Manifold Approximation and Projection. Manifolds are kind of like surfaces. And what it says is, hey, let's assert that on this surface, all of our data is uniformly distributed. Wait a second, this doesn't make sense. If it was uniformly distributed, that circle on the left should hit 2 points. That circle on the right should hit both 2 points. Or it should only hit 0 points. Well, the thing is, manifolds aren't exactly surfaces. One of the properties that manifolds have is that locally, distance behaves normally, behaves as you expect, but across large portions of the manifold, the meaning distances change. Distance from one point to another, from one point's perspective, is not necessarily the same as the other point's perspective. If you're familiar with some analogies used to describe general relativity, and if you're intimately familiar with general relativity, I know you're going to cringe at me. The rubber sheet analogy, where they take a giant bowling ball and drop into center, that's essentially what UMAP is doing. It's saying the density of the data set at that region of space kind of compresses space and makes things that are close in ambient space appear farther away, which is kind of the logical inverse of general relativity, where it compresses space and makes them closer. Of course, I could be reversing that part. Long story short, this is what it's saying the data is. This is uniformly distributed. It's just that the surface itself is a bit more malleable than what we think of. And wait a second. That means if these are uniformly distributed, these circles are the same distance, they're both hitting 2 points. Oh no, not again. Yep, that's where the nearest neighbors graph comes in here. Except instead of just being optimization, it's okay, from mathematical theory, how do we get here? And from here, we can actually build up a probability distribution that directly makes sense, isn't just straight up asserted. And in this case, the probability that X has Y as a neighbor, so in other words, the edge going from X to Y in the nearest neighbor graph, like the probability that that edge really exists, they're really neighbors, is can be shown to be this this exponential distribution, which note there's no divisor that we need to calculate or even approximate. It's just a single relatively constant time number that we can get. However, this is a directed graph. And so one of the things we do need to resolve is the fact this is a directed graph, or more closely to the manifold, the fact that one point will say, hey, you're my neighbor. The other says, no, get away from me. Points will not agree on what is close to each other, what are neighbors. And so what UMAP does is, if we ignore the Boolean logic for a second, this is essentially the probability that either one edge exists or the other edge exists. So as long as one of them exists, we're good. For the lower dimensional case, it just ignores that and just says that original case assumes like basically an undirected graph, and just does the one probability for both edges. And this row, it's not constant in the high dimensional case, but it's constant in the low dimensional case. And that's the important part, because that's the case where we're recalculating distances and probabilities multiple times. And so there was a lot here. I can go over the wrap up now, and then you can ask questions, wrap up for UMAP and ask questions, or you can ask before you even start with this, because that was a lot. And there's even parts of the UMAP paper that has category theory, and oh God, category theory, get it away from me. So the thing that UMAP and tSNE try to preserve is what points are close to each other. It preserves local relationships. And it's actually pretty effective regardless where you're starting from and where you're going. So going from 784 to 50, or going 50 to 2, it's still relatively effective. You're not going to lose that neighbor relationship, that information. But the space itself that you end up with really doesn't have any physical meaning. It's not like you can say, oh, this means that it's these and these genes that have effect. As convoluted and hard as it is to tease out in PCA, impossible here. And so UMAP is much better at clustering things. But before we go to code, any questions?
I have a few questions. I guess it's not really like the mathematical formula part, because that's something that's really beyond me. Like, I have the stereotype, biologist, math scares me. It actually really does. So I'm really interested in like the interpretation of, applying UMAP and what can we get out of it that's different from PCA. So Connie kind of asked this question earlier about, once you plot it. So for example, what you have this plot that you're sharing right now. So if you say that the distances are somewhat preserved in UMAP. So if we take.
Oops, sorry. Bad habit of interrupting on my part.
Oh, okay. So I understand clustering, like within a cluster, things are more similar to one another. But what about between clusters? So if we say, like, take that little cluster in the top left, and we compare it to the darker blue cluster on the right, but then there's a cluster that's somewhat closer right below the smaller cluster. So does that mean that smaller cluster is more similar to the cluster below it and less so than the one further away? Or can you actually only interpret within a cluster, like anything within this cluster is similar, but you can't then like kind of compare across clusters?
So yeah, you can't really compare across clusters. There are like a couple of what may be clusters that are kind of joined at a point, or at a point when it's a bunch of points. But so there may be something there where, like, okay, these kind of seem like on the left here, these might be two clusters that have portions that are close to each other within the clusters. But like this one and this one, yeah, like that's one of the things you lose in UMAP. With PCA, you could say something about stuff that ends up all the way over here and stuff that ends up all the way over here. Oh yeah, there's like this physical difference, but these two clusters may have more in common than these two. The thing it tries to preserve is strictly local distances, things that are already closely together to begin with. Things that are far apart to begin with, forget about it. You keep nothing.
Okay, that's really helpful to know. Thank you so much.
Yeah, it's very much what's close, stuff that's close stays close. And one of the things that can actually happen with the k-nearest neighbors graph part is that you can end up with, instead of a single interconnected graph, you can have like 2 or even 3 graphs that are completely disjoint from each other. The mathematics still holds in that case, but these are kind of like they're disjoint from each other. And when projected down into this lower dimensional space, completely separate.
Yeah, that makes sense to me. Thank you.
Is there, all right. And in my case, more specifically to like what my interest was with this was the thinking of, oh, I'm trying to take these blocks of data points and figure out, like, see if they're actually close to each other. And so that's kind of like the motivation for me with UMAP, was seeing, okay, how many of the lines do I end up connecting things end up pointing within clusters and not across clusters? And so that's like more like where I was coming from, like what I cared about, and what I'll share what my takeaway was at the end from this when I actually get to that image. But like that's what I expected UMAP to tell me. Okay, there's actually something going on here with blocks being close to each other, actually being close to each other or something. Any other questions though? With that, okay, so this part here, you do need the UALT library. UMAP's has no built-in functionality into R. And before I actually get too distracted, I did mention PCA/SVD is built into R. There may be other versions of it that are available through different libraries that may be more optimized. If I remember correctly, I think the version built into R is a serial implementation. The SVD is not too hard to make parallel. So there's probably an implementation out there that is, if the built-in one isn't. But UMAP, you definitely need the UALT library. That's the implementation that the authors of the UMAP paper have for R. And in this case, you don't always get the actual full embedding model. In my case, I wanted to show both this clustering behavior, but also projecting the points onto the same, apply the same mapping to it. And in order to get that, you do need to explicitly ask for it. And we end up with this embedding, which once again, you can't really just multiply with it. You do need to use UMAP transform, but you can take that model and apply it to new data and transform it. And that's how you get the dimensionality reduction for applying this model to new data. That's how you use UMAP for that. But normally I don't know exactly what the rest of here you do or don't get, but in this case, the primary thing you get back out of it without that is just the actual the embedding model itself, or the embedding itself. This is the actual like data points in the lower dimensional space, and that's the important part of what you get out when you don't specify this. And so yeah, applying UMAP to the data, then UMAP transform to the centers of mass, the centroids, to get the transformed version of that. And what I do have for this one is a plot that has the centroids projected onto space, but also I've drawn the lines of for each centroid represents a chunk of my data, and draw the lines between it and the blocks that have the most neighbors for the points within that block. In other words, I'm a block, I have 500 points. This block has like 400 neighbors with my points, type of thing. And so this is the one that took quite a while. Brief aside, I couldn't really find anything that was really appropriate for this type of visualization. And this is explicitly one of these things that you should avoid doing this exact type of thing. I'm only showing 3 lines. If this was 5, it would Stack Overflow. This is actually like, so I do want to point out this code here is super jank. But fortunately, it's going to take forever. And I forgot this part takes forever to just show this image.
Since we all, or most of us work in R, we can say that we feel your pain when it takes forever to load a plot. We totally understand your pain.
So yeah, in grad school, I worked primarily in Python. That's why I don't work in Python anymore. Granted, okay, so a bit of a connection here. PyNNDescent is an implementation of nearest neighbor descent, but that was written by one of the authors of UMAP, and that's actually the k-nearest neighbors algorithm that's used in UMAP to do the whole k-nearest neighbors part. And Leland McInnes gave me the idea of basically re-implementing that in C++. One of the things that the R version does not use PyNNDescent because it's actually implemented in C++ and it uses a different algorithm for the k-nearest neighbors graph. But the PyNNDescent, that actually uses a JIT to get optimized and actually run fast. And like the main things you get, in this case, the main things you get out of re-implementing in C++ isn't necessarily the raw speed of an interpreted versus compiled language. Yeah, in that case, you might have to compile it and then run it. When, if it's written C++, I can just hand you the binaries. But the main part of the motivating factor for re-implementing in a different language is just, what does the language give you? I get to play with raw memory. Don't necessarily want to play with raw memory. It can be scary at times. Okay, so, okay, there we go. Let's see if I can expand this to get a bit of a clearer view of this. Don't tell me this is going to take forever just to resize it.
That's due to the fact that this is a ggplot. So you have a lot of points on that ggplot. And so ggplot's really not meant for this.
Yeah, no. There is like graph visualization stuff, but not stuff where the space actually matters. It's more like, oh, this is like strictly graph representation where this is just representing arbitrary connections and relationships and whatnot, where the points have no necessary spatial meaning. And so I needed the spatial meaning in this case.
Oh yeah,
okay.
No, I'm not picking on your plot at all. I'm just pointing out that this is a lot of points for a ggplot and it's really not meant for that.
Mm-hmm. Yeah, that's kind of like, I recognize that you saying that probably know why I end up doing this. That's more for, beyond just the person who's going to know like why I ended up down this. And I think like ggplot is under hood. I'm not exactly sure what the memory model of R is, but I know a lot of it is basically interfacing with C code, and it might be a C, maybe C, or in the case of UMAP, that's C++. you can interface with C++ from C. Telling myself that but yeah, in this case, it might just be taking like all these lines that's being drawn and representing them on the stack. And
it is. And so I was just pointing it out that if you work with these types of plots a lot, you may want to use a different package than ggplot. But obviously this is the built-in plots for it. So yeah, no, it was just a point of note for anybody on the call that might do this a lot. If you don't want to wait for them, you may want to use a different plotting functionality.
But yeah, so but to go back to the plot itself, there are lines going across clusters, and that's fine. But there's also a great number and like a great density of them that are within clusters. And like, I didn't need it to be perfect. I just needed to be like, okay, I get something out of doing this. And this was like what sold me on going down that route and trying to, instead of just doing random initialization, and to actually try and compare like things to each other. And this was like, aha, I'm confident in what I'm doing now, only slightly, but something. And so that's like, this was not just a random example. This was like, I wanted to show something that had meaning to me. And this was something that gave me a lot of courage in what I was doing. And so, but yeah.
That's very cool. That is awesome, real result.
And like, I think this is still the image that I use on that repo for the code that I'm working on. It's still an ongoing monolith of a project, partially because A, I'm insane, and B, kind of hard to work on something I'm not being paid for when I also have to be paid. But it's slowly ticking along, and this is like the thing that represents what I'm trying to do. So any other questions?
I have another question. With so I haven't really played around with UMAP very much, but from using PCA plots, like you can kind of say like your component 1 explains this many % of your variant versus 2, and you can kind of generate that kind of data with UMAP. Is something similar available? So I see that you have like embedding 1 and 2, so I'm assuming that's some sort of dimension that's being plotted there.
Okay, so it's been a while since I've looked too closely at the actual math and theory behind UMAP. So some of the exacts are not coming to mind for me right now, but one of the things I do really want to stress and really want to emphasize is that these axes, embedding 1, embedding 2, they themselves might as well mean nothing. It might as well not actually represent something. And there's cases where that's kind of one of the drawbacks about nonlinear methods, where what you're preserving isn't necessarily like the ambient space in like the case of genomics, like different effects of different genes or different upregulations of different genes. In this case, the actual two-dimensional space itself as for where points end up relative to each other, that is a lot more meaningful. That has a lot more significance. And I really would my advice is I would avoid thinking about these axes as this point is here because it has this amount of this effect and this amount of that effect. This point is here because its friends are here. It's around good company, to speak metaphorically.
Okay.
was actually kind of as a side to that, I was gonna ask if, because I'm not sure what the group's made of, if there are a lot of biologists here, but I was gonna give an example that is more biologic to test that I'm understanding the interpretation between these two. So like when we talk about PCA, like you were saying, Yuka, like it's really easy. You can say this much of the variance is explained by this component and then this much by the next. And if you see two clear clusters, you can say, like if it's two clusters like sex, you can say one's higher in expression than the other, or they're very similar to each, or they're different in the same ways, kind of a thing. Like the genes are different in the same patterns. Whereas with a UMAP, you wouldn't, like, I don't think of it as comparing across like that. So you would do something like getting cell types. And so all the points that are together in the UMAP, you'd want to see what cell type they are, but different points that are farther apart in the UMAP, you wouldn't compare an expression from the UMAP. You would have to like get a cluster label out of the UMAP and then compare expression.
Yeah, I've seen a lot of UMAPs being used for single cell genomics. So I figured like that's kind of the better application, is to, label, tumor cells versus normal cells and like different types of cells. But yeah, I was just trying to understand, what does it mean when it's far away from each other? But if there is no meaning, then that's
Okay, yeah, because I wasn't sure if what I was explaining was right. I didn't mean to come across that way. It was just that's how I had interpreted it. So I wanted to make sure that was okay.
Yeah, that makes sense. And in this case, I don't have readily on hand, but like what this is trying to do is show like different classifications. In this case, because this is MS fashion, like the shirts end up in this one cluster, the pants end up in this other cluster. And because I'm not classifying in that way, some of these clusters will be kind of merged and have more than one article of clothing associated with it, or but like some of these clusters are going to have, this is literally just shirts type of thing. And so it's more teasing apart classifications from each other, not where they ended up, where they were in the original space. And yeah, like I do have a bit of a background applying math to biology, but a completely different like world of math essentially. So I don't have a lot of good biology examples from experience, despite applying math to biology. It's more, in my case, it was more, how do we model this kidney tissue from no data? Not literal no data, but might as well be no data compared to some of the data sets you would use this stuff for.
Do we have any other questions for Anabel? Doesn't have to be from biological standpoint.
Doesn't have to be strictly related to just this. If I can answer it, we have honestly plenty of time.
And so if people want to reach you after this workshop, if they have any other lingering questions, what would be the best way?
Let me just type my email into the chat. This is my email is the best way to reach me. It gets pushed to my phone. There are emails I do end up forgetting I get, but I'll try not to. I'm not the best at remembering things, to put it lightly.
Okay, so if there are no more additional questions, let's give Anabel a virtual round of applause, or you can unmute and actually do that. That's fine too. But yeah, thank you so much, Anabel, for such a great talk. It's very in-depth. I think I've learned some math today, which I didn't really realize I would. So thank you so much. And so we've been recording this workshop. So if anyone wants to refer back to it, we will have this up on YouTube in the next 2 weeks. So keep